Hey BioTeam. So a point mutation is any mutation to a gene or RNA strand that changes just a single base in the genetic code. So we'll go through a couple of examples of point mutations here. Let's say that we have a code of messenger RNA that's been transcribed from a gene. Now you guys have learned that every 3 bases of this RNA strand equals a codon for a single amino acid. And so biologists will often add these codon frames in order to distinguish the codons more easily. So this particular RNA strand would code for 7 total amino acids before reaching a stop codon. That is, it would translate into a protein that's 7 amino acids long. But let's say that this strand of mRNA undergoes a mutation. Let's say it undergoes a substitution, which occurs when a single base in the RNA strand is replaced. So in this substitution, a cytosine has replaced an adenine. Well, as a result of this substitution, the protein that gets translated from the RNA strand will be slightly different. Now, this codon, guanine, cytosine, cytosine, codes for alanine instead of aspartic acid. So this could affect how the protein folds, especially because the original amino acid was a polar amino acid, but the new amino acid is nonpolar. So that's a substitution. Another type of point mutation is something called an insertion, which occurs when a base is inserted into the code. So in our insertion, a cytosine has been inserted into this second codon, and all of the other bases shift down as a result. So in terms of the consequences of this mutation, our first codon was unaffected. It's still AUG and still codes for the amino acid methionine. But all of the remaining codons that are downstream of this insertion are affected, and the amino acids will be significantly different from the original protein, including now a stop codon that has been introduced into the sequence earlier than in the previous protein. So now there's only 6 amino acids in this protein instead of 7. So that's an insertion. The opposite of an insertion is a deletion, which occurs when a base is removed from the genetic code. So in this deletion, an adenine will be removed from the second codon, and all of the other bases will shift back as a result. So similar to the consequences of an insertion, with this deletion, the codons before the mutation are unaffected. They'll code for the same amino acids. But all of the codons downstream of the mutation are affected. So both insertion and deletions are called frameshift mutations because they're mutations that break up the original codon triplets. That is, they shift the reading frames of the codons so that the original triplets are no longer together. And so any insertion or deletion of a number of bases that's not divisible by 3 will result in these codons being broken up, and new codons will form as a result. That's it. And at this point, you guys have some practice problems. We'll see you next class.